So gene expression involves the copying of the genetic material in a mechanism known as transcription. This copying mechanism is distinct from replication that I just mentioned a minute ago. Transcription is a process in which RNA molecules are synthesized on a DNA template. So in this instance, what you see, or what happens during transcription, is genes get copied on an individual gene by gene basis to produce a functional RNA molecule. That RNA molecule can then go on to do different things or have different functions inside of the cell, depending on the class or category of RNA that has been produced by transcription. Most genes are considered protein coding genes, and that means that the RNA that is produced is used as instructions to synthesize a protein. That process of using RNA as the template and synthesizing protein based off of the instructions in the RNA molecule is known as protein synthesis or translation. What I typically like to do before I delve too deep into molecular genetics or gene expression is to talk a little bit about the historical perspective that led us to our understanding that DNA is in fact the genetic material. So we'll start off with the work of Fredrick Griffith. And Fredrick Griffith was a physician who didn't actually really like working so much with people. He was interested in more of the science behind medicine. He happened to work in a surgical hospital where he had access to bandages quite frequently, and bandages contain pus, and pus is the result of white blood cells. Now, it turns out that these white blood cells have very large nuclei and relatively little cytoplasm. And Fredrick Griffith was really interested in studying the nucleus of the cell overall. He was able to isolate nuclei, and he found or determined that within the nucleus of a cell was an acidic substance that he referred to as nuclein. Next, we have the work of Phoebus-Levene. Now, Phoebus-Levene first determined that DNA contained equal amounts of similar molecules that he referred to as nucleotides. He then studied the nucleotides in a little more detail biochemically and was able to establish that there was in fact a carbohydrate component to every nucleotide. He also identified that as a 5-carbon sugar known as a pentose. And then more specifically, he identified the sugar as deoxyribose. One of the things that Phoebus-Levene proposed was known as the tetranucleotide hypothesis. He proposed that DNA was a linked series of 4 nucleotides, and the order of the nucleotides was always the same. So currently, you hopefully have a good understanding that there are 4 distinct types of nucleotides that are abbreviated by the letters A, C, G, T. What Phoebus-Levene proposed in this hypothesis was that DNA was a linkage of these 4 nucleotides repeating over and over and over again. For example, A, C, G, T, A, C, G, T, A, C, G, T, and so on. Next, we'll talk a little bit about the work of Fredrick Griffith. Now, at this point in our understanding, there was a huge debate in the scientific community as far as what specifically was the genetic material. Nuclei were known to contain DNA as well as proteins. The 2 camps of thought was that proteins were much more complex molecules than DNA because proteins, as you know, are made up of 20 different amino acid monomers, whereas DNA only has 4 monomers or building blocks known as nucleotides. So the argument was that proteins were much more complex compared to DNA, and something as important as the genetic material was most likely a very complex type of molecule, like a protein. So Fredrick Griffith's work specifically focused on looking at different strains of bacteria associated with pneumonia. There were 2 strains he was working with. One strain we will call the R strain, which stood for rough because under the microscope they had a rough appearance. And it turns out that this rough strain of this particular bacterium was harmless, meaning that it didn't make individuals very, very sick. The second strain is known as the S strain, and the S stands for smooth because, again, under the microscope they had a smooth appearance. This particular strain actually was extremely pathogenic. It caused extreme illness in individuals that were infected with this particular strain. Now, Fredrick Griffith conducted a number of experiments. The very first experiment or series that he conducted, he utilized the S strain, and what he found was that if he injected the S strain into mice, the mice would die. So from this, he concluded that, again, this was a truly virulent strain, extremely pathogenic, eventually leading to the death of the mice. In addition, he also tried utilizing the R strain, and the R strain, once he injected it into the mice would get a little bit sick, but survive overall. And so in this particular case, this is how he determined that this was a harmless strain. He then took his experimentation one step further, and he utilized a technique where he heat treated the S strain. Now remember, the S strain in panel A would cause the death of the mice if the mouse was injected with this particular strain. Because he wanted to test whether DNA or proteins were the genetic material, he heat treated the S strain, because knowingly, when you heat treat proteins, they denature or fall apart and become non-functional. So by heat treating the S strain, he was hoping to make the proteins become non-functional. And in this case, what he found when he injected the mice with the heat-treated S strain was, in fact, the mice would survive. The last experiment that he did was he decided to take the heat-treated S strain and mix that heat-treated S strain with live R strain. He took that mixture, injected it into a mouse, and what he found was that the mice would die. In addition, when he analyzed the bacteria that were present in the dead mice from this particular experiment, he found that there were, in fact, virulent forms of the S strain present. His conclusion from this set of experiments was that something from the heat-treated S strain actually transformed the R strain into becoming virulent and eventually led to the death of the mice. From this, he proposed what is known as the transforming principle. And the transforming principle simply states that something from the S strain transformed or changed the R strain to become virulent. Now, Now, what that something is wasn't 100% clear in this set of experiments. However, because the proteins were in fact denatured, he hypothesized that DNA must be the component that is allowing for this transformation to take place. The next set of experiments come from the work of Avery, MacLeod, and McCarty. In this particular set of experiments, they worked with the same two strains of bacteria that Fredrick Griffith did that we discussed in the previous slide. However, in this particular case, what they did was they heat treated the S strain, and then they began a series of biochemical extractions to remove carbohydrates, lipids, and a majority of the proteins. When they did this, they used that extract that was prepared and mixed it with live R strain. And it turns out that they were able to transform the R strain, similar to the way Griffith did that we just discussed in the previous slide. So their conclusion here was that something from the S strain, after the extraction of carbohydrates, lipids, and proteins, was allowing a transformation of the R strain to take place. They then took their experiment one step further. They heat-treated the S strain. They performed that series of extractions, removing carbohydrates, lipids, and proteins. However, this time they also treated it with an enzyme known as a protease. Now, a protease is an enzyme that will destroy or degrade any remaining protein that might be in their sample. So at this point, we are eliminating all proteins from the extract. They mixed that extract with live R strain and also found transformation did in fact take place. They then took the experiment another step further. Heat-treated the S strain, performed the crude extraction, removing carbohydrates, lipids, proteins, treat with the protease to remove the remaining proteins. So what is left behind in the extract should only be nucleic acids. In this experiment, they took that extract and treated it with an enzyme known as a ribonuclease. Ribonucleases are a family of enzymes that degrade RNA. So now the RNA has been removed from the extract. They mix it with the live R strain and found that transformation did in fact occur. Now at this point, you may think, well, they've been able to demonstrate then that DNA is the transforming component because that's the only thing left in their extract. However, they did take this experiment one step further. Heat-treat the S strain, perform the crude extraction, treat with the protease, treat with the ribonuclease, and now treat with a deoxyribonuclease. Deoxyribonucleases specifically degrade DNA. Now their extract no longer contains DNA, and when they mix that with the R strain, they found that no transformation took place. From this series of experiments, they concluded that DNA must in fact be the genetic material. So the last set of experiments that we discussed by Avery, MacLeod, and McCarty suggested that DNA was in fact the genetic material. However, those experiments were all in vivo, meaning occurring in the test tube. And so there were some, again, those in the protein camp, that still thought that DNA could not be the genetic material because it was too simple, even though we had this compelling evidence from the last set of experiments. The final set of experiments that allowed us to completely understand that DNA was in fact the genetic material come from Hershey and Chase. Now, Hershey and Chase were utilizing bacteriophage, And bacteriophage are viruses that specifically infect bacteria. They were working with a phage known as T2, and they knew a little bit about the infection process. They knew that T2 contained approximately 50% protein and 50% DNA. This particular virus is in fact a simple virus made up of a protein coat and the genetic material DNA inside. They also knew that this particular bacteriophage infected via injection of the DNA, meaning the bacteriophage would land on the cell surface, inject the DNA, and then the cell's machinery was taken over to produce more viruses. So what they did in their series of experiments was to take the bacteriophage T2 and grow it in two separate cultures. In one culture, they exposed it to a radioactive component of sulfur known as 35S. In a separate culture, on the right-hand side of your figure, they grew the bacteriophage in a culture containing radioactive phosphorus known as 32P. They allowed for the viruses to infect bacteria, and then they isolated the bacteria to analyze the bacteria that were present in either case. Now, before analysis, they then took their cultures and put them in a blender. What the blending does is cause a physical disruption between the protein coat that is on the surface of the cells and the cells themselves, hence uncoupling the protein component of the virus from the actual cell that's being infected. Because the argument was that if the protein stays present, even though we know the DNA is getting injected into the cell, that protein is still present and perhaps could be contributing to genetic information. So, by putting these cultures in a blender that shears or cuts off the protein coat from the bacterial surface. Once the blending was complete, they were then able to analyze the bacteria in each one of the cases. They analyzed the bacteria grown in the radioactive sulfur culture, and we should take a step back and say that by incorporating radioactive sulfur, they were adding a marker or a beacon or a way of identifying proteins downstream. So by looking at the bacteria present in the culture that was originally grown in the presence of radioactive sulfur, they analyzed the new viruses that were produced and did not find any radioactive sulfur component. However, when they analyzed the cells and the bacteriophage isolated from the culture that was grown in the presence of radioactive phosphorus, and here we should take a step back and say that the radioactive phosphorus would specifically allow us to identify DNA because it would get incorporated into DNA. What they found when they analyzed those viruses was that radioactive phosphorus was in fact present in new viral particles. What's more, they could then allow those new viral particles to infect new cells and saw the continual transfer of the radioactive phosphorus from generation to generation of new viruses. This then was the last bit of evidence that indicated that DNA was in fact the genetic material and not proteins. There is also additional evidence for DNA as being the genetic material in eukaryotes. For example, the distribution of DNA when you compare the amount of DNA in a haploid cell versus a diploid cell. Here in a variety of organisms, you see that the haploid concentration of DNA is half that of the diploid concentration. And at this point in time, it was pretty clear in our understanding what was happening during the mechanisms of mitosis and specifically meiosis. So given that a diploid cell had twice as much genetic information as a haploid cell, followed along with the concept that DNA was in fact the genetic material. However, there were also examples from mutagenesis studies. Mutagenesis studies allow for mutation of genetic material to take place. Okay, now that we have a good understanding that DNA is in fact the genetic material, let's talk a little bit more about DNA structure. So we've already established from the works and our historical perspective that the building blocks or the individual units that make up DNA are known as nucleotides. So what I want to do is review nucleotide structure in a little more detail. Every single nucleotide, whether it is for DNA or RNA, contains a 5-carbon sugar known as a pentose sugar as a broad category. But more specifically, RNA nucleotides contain the sugar ribose, and DNA nucleotides contain the sugar deoxyribose. If you look at the two structures in this particular slide, you see that they are almost identical, with the exception of the structures or functional groups that are added at carbon number 2. So starting at the left-hand side, starting at the right-hand side, if you look at the right-hand side, the very first carbon is labeled as carbon 1, then the next one carbon 2, 3, 4, and 5, going in a clockwise orientation. So at carbon number 2, in ribose, there is a hydroxyl group that is present. And in deoxyribose, there is no oxygen present, hence the deoxy, meaning without oxygen, at carbon number 2. The exact and correct name is 2-deoxyribose because it's the number 2 position that is missing the oxygen. In addition to having the 5-carbon sugar, every single nucleotide also has a nitrogen-containing base referred to as a nitrogenous base. There are 2 broad categories of nitrogenous bases. In panel A, you see the generic form of what is known as a pyrimidine. This is a 6-membered ring structure. And below that, you see the generic form of a purine. And a purine is a 5-membered ring fused to a 6-membered ring. Specifically, there are 3 different types of pyrimidines: cytosine, uracil, and thymine. And there are 2 types of purines, guanine and adenine. Now, I won't ever ask you to recognize these specific structures, but you should have a good understanding that a pyrimidine is a 6-membered ring structure and a purine is a 5-membered ring fused to a 6-membered ring. You should also know which specific bases fall into which categories. Cytosine, uracil, thymine are pyrimidines, guanine and adenine are purines. In addition to the 5-carbon sugar and a nitrogenous base, every single nucleotide will also have at least 1 phosphate group. In the figure that you see here on the left-hand side is a complete nucleotide with the base starting on the right, the sugar in the middle, and then in this case 3 phosphate groups linked one right after the other with covalent bonds. The term nucleoside is a molecule that is just a sugar and a base. And the term nucleotide is a very generic term that refers to a molecule that has a base, a sugar, and at least one phosphate. However, the building block for DNA and for RNA will always contain 3 phosphates like you see in the figure on the left-hand panel. On the right-hand panel, you can see 2 nucleotides that have been linked covalently to one another. And this is in fact how DNA and RNA is built, by adding nucleotide to nucleotide to nucleotide over and over and over again. Both DNA and RNA molecules are polymers. They are polymers of units known as nucleotides that are linked one right after the other. Both DNA and RNA have what's known as a polarity to them, meaning there are distinct components on either end of the molecule that allow us to recognize, identify, and specifically refer to one end of the molecule or the other. For DNA, this polarity or directionality is referred to as the 5' end or the 3' end. The number comes from the sugar that's present in the nucleotide for an individual nucleotide. Again, the sugars are 5-carbon sugars, and if you go back to the slide looking at just the sugar, we went through and discussed carbon number 1, carbon number 2, carbon number 3, carbon number 4, carbon number 5. However, these carbons also have the prime designation to differentiate those carbons from the carbons found in one of the nitrogenous bases. So anytime you see the number with the prime, that should clue you in that we're talking about carbons in the sugar itself. So for every single nucleotide, there is a 5' carbon and a 3' carbon. In this example, in the figure, you see 3 nucleotides linked one right after the other. There are 2 things that should clue you in that this is a linkage of DNA, and that is because the number 2 position on each one of these sugars is missing the oxygen, hence deoxyribose. And also, we haven't discussed yet, but in DNA, there are 4 distinct bases, A, C, G, and T: adenine, cytosine, guanine, and thymine. In RNA nucleotides, there are also 4, A, C, G, and U. There are no T-containing nucleotides in RNA. It is replaced with uracil. So in this structure that you see in the figure, there is a T for thymine, which then should also clue you in that this is a DNA molecule. Okay, so back to the polarity or the directionality. At the top of this figure, you can see the nucleotide where the 5' carbon is of that top nucleotide. It is not covalently attached to another nucleotide. This is referred to as the free 5' end. At the bottom of this molecule, you see the last nucleotide in the structure, and here you can see what's known as the 3' end, meaning that the 3' carbon is not covalently linked to another nucleotide. So in this figure, the 5' end is at the top and the 3' end is at the bottom. Okay, so now that we have a good understanding of the building blocks necessary to build either DNA or RNA, again, a nucleotide. What I want to talk a little bit about now is the more complex structure of DNA and RNA. So we'll take one step back and we'll talk a little bit about the work of Erwin Chargaff. Erwin Chargaff was a biochemist and was interested in studying base composition and nucleotide structure in DNA. What he was able to do was to isolate DNA from a number of species and analyze the composition of the bases in those DNA samples. So if you look at the bottom portion of this slide, you can see he's got several organisms that he isolated DNA from. And what he found in analyzing the base composition was that the base composition was different from organism to organism. In addition, you can look and see the specific base composition, A, T, G, or C, and what he found in a variety of organisms that it was not in fact the same all the way across the board. This then does not support the tetranucleotide hypothesis proposed by Phoebus Levine at the beginning of the lecture. This suggests that depending upon the species, the base composition can vary, again, from species to species. What he did, however, find was that when he made comparisons, that is the ratio of A to T or the ratio of G to C, that was the same regardless of the species he isolated the DNA from. So while individual base composition varies from species to species, the ratio of A to T and the ratio of G to C was roughly 1 in every species he analyzed. Lastly, you can put the A plus G concentration or content and compare that to the C plus T concentration or content. Here again, the ratio is 1 to 1. So this again does not support that tetranucleotide hypothesis and suggests that there's some specific combination that occurs between the nucleotides to allow for these ratios to be found in every species he analyzed DNA in. What you see in this figure is actually what is proposed to take place between bases in DNA structure, and that adenine will bond or pair up with thymine, and guanine will hydrogen bond or pair up with cytosine. This is actually referred to as sequence complementarity, and that concept will come up over and over again as we continue to talk about gene expression. So then let's talk a little bit more about the structure of DNA. And as you probably are all familiar with the concept that Watson and Crick are the scientists that were credited with discovering the structure of DNA. However, they did use a lot of additional information from other scientists to get to this conclusion of the structural features or characteristics of DNA. They utilized the work of Erwin Chargaff that we just talked about in the last in the last slide. And in that slide, again, the concept of sequence complementarity comes into play with those really interesting ratios that Erwin Chargaff established. And that the concentration of A was always equivalent to the concentration of T, and the concentration of C was always equivalent to the concentration of G. This suggested that there was some way that those components interacted with one another. They also had the work of Rosalind Franklin. Rosalind Franklin was a female scientist who was very good at a technique known as X-ray crystallography. X-ray crystallography involves formulating ideas about structure based on crystals that have been formed of the molecule in question, and using X-rays and the diffraction pattern of the X-rays to then mathematically model what the structure of DNA was. Now, it turns out that Watson and Crick had access to all of Rosalind Franklin's data without her knowledge, and they used her data to support their idea or their hypothesis of DNA structure. What they proposed is the structure of DNA that we know today is the classic double helix molecule, in that it contains two molecules of DNA that interact with one another via hydrogen bonding of the bases and is in a helical shape or structure like you see in this figure. More specifically though, if we unwind the helix, we can look at the structure in more detail. Here in this figure, you see the two strands of DNA that come together to form the helical structure. This figure is a great figure because it shows an additional component that we haven't discussed yet, and that is that the strands are referred to as being anti-parallel. They are going in opposite directions of one another. The strand on the left-hand side has the 5' end at the top and the 3' end at the bottom where the tip of the arrow is. The other strand on the right-hand side has the 5' end down at the bottom and the 3' end up at the top. The two strands of DNA, again, are referred to as being anti-parallel, meaning they are going in opposite directions of one another, very nicely depicted with the arrows that you see in this figure. So if you take this structure and twist it up, you get the helical shape that we know as the classic double helix. Also very nicely down the middle, you can see the interaction of the bases of the two strands and that specific base pairing that we just talked about, that C's will always hydrogen bond with G's and A's will always hydrogen bond with T's. This is known as the base pairing rule or sequence complementarity. In addition, we can talk a little bit about RNA structure. And RNA structure is similar to DNA structure in that it is a linkage of RNA nucleotides, one right after the other. It is, however, in most cases, a single-stranded molecule, which is distinct from the structure we just discussed for DNA, which is a double-stranded helical shape or structure. There is a polarity to RNA molecules as well, a 5' end and a 3' end. And in this figure, the 5' end is at the top, the 3' end is at the bottom where the tip of the arrow is. We will discuss different classes of RNA in one of our upcoming lectures, as well as non-coding RNA in one of our upcoming lectures. The last thing I want to touch on with this particular lecture is that DNA in the eukaryotic cell is not found simply crammed into the nucleus. There is a very complex organization of the genetic material in the nucleus of the eukaryotic cell. So in this figure, you have some idea of that complex organization, in that at the very top of the figure, you see the double helix, what I typically refer to as naked DNA. It's just the DNA by itself. However, again, we know that DNA is in complex association with proteins in the eukaryotic nucleus. Those proteins are a family of proteins known as histones, and those histones allow for the condensation of the genetic material and the organization of that genetic material. So instead of taking lots and lots of DNA and just cramming it into the nucleus, there's a complex way that the DNA is organized to be able to fit compactly in the nucleus and to be able to organize that genetic material. And I think that's really all I want to say with this particular slide, just knowing that there is this complex organization with the proteins that take place.